So thank you, Henrik and Paul, for inviting me. I was amused this morning when Jean-Pierre was talking about how he was introduced to neurofilaments. I think those of us who were in the field for a long time, many decades, that the way we got involved was very different than how things turned out. I was interested because I was interested in Alzheimer's disease, and at that time, Mike Shalansky, who was a colleague of mine, had reported that neurofilaments were the core of neurofibrillary tangles, and so we thought we would study the biology of these neurofilament proteins, only to come to find that they weren't the core. Tau was the core, and then neurofilaments were dead in the Alzheimer field for a few decades, and then they've slowly reemerged as players, both in terms of being part of the tangle and being hyperphosphorylated in Alzheimer's disease. And I think the ups and downs of the field, and we've seen 3 or 4 different ups and downs. It's now going to take an up phase, both with the biomarkers, and I think the roles that neurofilaments are going to be playing in synaptic function are going to have some ramifications that will be very surprising beyond what I'll be talking about today. So he also mentioned the fact that he was told that neurofilaments were a contaminant in his synaptic preparation, and this was part of the thinking for a long time, that this idea that the bulk of neurofilaments move down axons and the entire cytoskeleton was being chewed up at the end, and that there would be no neurofilaments in a synapse, really colored the way that people looked at neurofilaments in synapses. And all these early proteomic studies would say, well, we've got neurofilaments there, we must be contaminated. And slowly over time, the more sophisticated proteomics over the past 5 years or 10 years have realized that, in fact, they always see neurofilaments in purified synaptic preparations. And that's one of the pieces of evidence that was slowly trying to convince us that we were, some of the things we were interested in with regard to neurofilaments at synapses actually were true. So I'll talk a little bit at the beginning about that as to how we started to realize that synapses were important with respect to neurofilaments. So let's see, which one of these is the red one?
No, it should be probably that one.
Yeah, it's not. Oh, okay. So much of the work that I'll be talking about relates to 2 papers that were published, one in 2015, Molecular Psychiatry, and one very recently in Translational Psychiatry. And the senior author is David Yuan, who's really spearheaded a lot of the neurofilament work. And we have other interests in endosomal lysosomal function, and he's taking this on with another group that has been in the field for a long time, Mala Rao, Varana, who's now retired, who have in their own right published very interesting work at NKI and NYU. and before that at various institutions. And then, of course, Jean-Pierre has been instrumental because a lot of the experimental models that we created originated with the knockout of individual neurofilaments that he generated. So he's been a long-standing collaborator. So I'll briefly touch on each of these topics. There are certainly basic properties that have led us to the synapse that I'll talk about, and then very briefly talk about the first evidence, solid evidence that we had that functionally neurofilament, and specifically the NFM subunit, had very specific role in synaptic function, regulating the D1 receptor. And then talk about the more recent work that was published very recently on NFL and its role in regulating NMDA activity. And then touch at the end on metabolism, which is still an ongoing study in our lab, but we have some interesting results to report. So Jean-Pierre mentioned this morning that for many decades, actually, the central focus had been on peripheral nerves and on the fact that neurofilaments support the caliber of large caliber myelinated axons, and that when you reduce neurofilaments in any variety of ways, you reduce the caliber. And it was also thought that there was the central dogma, as I mentioned, that the neurofilament lattice moved down like a tube of toothpaste and got chewed off at the end. And for a number of decades, there was a debate about whether most of what you see in the axon is stationary lattice or whether it's a continuously moving structure. And this kind of dissuaded people into thinking that the synapses are just a place to chew neurofilaments. In the CNS, on the other hand, neurofilaments don't seem to have much of a role in regulating caliber. Even in myelinated axons like the corpus callosum, they're not huge axons compared to peripheral axons, but they are significantly large and represent one of the larger populations in the CNS. But if you knock out neurofilament, as we did with an HM knockout, you remove as much as 90 or more % of the neurofilaments, you have virtually no effect on the caliber of these axons. And this is, if this is a pointer, which apparently it isn't, you can see here that the knockouts have basically the same cross-sectional areas of the myelinated and unmyelinated axons, even though they have very little neurofilament protein. And the same goes for mouse and monkey that someone else had, Pashko Rakesh had reported later. So without going into the details here, this shows a number of the other features of neurofilaments which were always puzzling to us, which is why all other intermediate filaments are either homodimers or heterodimers of 2 subunits. Why do neurofilaments have 4 subunits? And even more importantly, why do they have this very elaborate phosphorylation pattern where they have phosphates on the head domain and they have even more phosphates on the C-terminus. And in addition, studies that we did a number of years ago showed that after synthesis, the head domain phosphates start to change. They turn over differentially as neurofilaments enter the axon and turn over. So there's both a complex number of phosphate sites phosphorylated, but then they're turning over for reasons that have nothing to do with assembly. There are some that are related to assembly, but this whole notion as to why there are 6 different kinases that modulate neurofilament proteins and all these sites, it just didn't seem to match with the idea that they're static structural supports for neurons and why there are 4 of them. So NFL in particular is, I think it's been mentioned that it is it forms the core of the neurofilament, and its assembly and the assembly of neurofilaments themselves seems to be regulated by head phosphorylation on NFL. If you remove those phosphates, you seriously alter the assembly of the filament. So those are pretty straightforward related to neurofilament formation. Curiously, David Yuan and our group, using different models of missing subunits, was able to show that the L subunit is not essential for transport. You can remove it and you will still transport the other subunits that still remain. This was an observation that was made 2 decades ago, and at the time it just had no meaning. It came out of the blue. NFL is phosphorylated in dendrites during LTP, and no one did anything, including us, with this information for 20 years. And I think you've heard about the mutations of NFL. I won't go into that any further. But what David showed some years ago was that there is a requirement to have at least 2 subunits in order to move a filament, or to move an assembly of neurofilament proteins. And one of those essential subunits is M, and the other is internexin. And if you remove internexin, you can substitute L, but L combined with H will not move along axons. So what this did was tell us that even though neurofilaments are usually depicted as assembled filaments, that they can move in these small assemblies. And we can demonstrate small amounts of neurofilament moving in this soluble heteroligomeric form. And we think those are the ones that ultimately can move faster and move into the synapse and regulate in a more rapid way changes in neurofilament formation. It's also the case that neurofilaments, as Jean-Pierre mentioned, are regulated by TDP-43. We know that there's TDP-43 in the synapse, and that it's likely that there's local synthesis of neurofilaments in synapses. So this is just another confirmation of the fact that without NFL or H, you can get transport of oligomeric but not filamentous neurofilament assemblies. And this becomes very important when we think of the synapse, where conventionally it was difficult to visualize filaments extending all the way into the synapse. And then this construct and another similar triple knockout in which you knock 3 filament subunits out, you have very little change in, or no detectable change in synaptic structure, but a profound effect on synaptic function. And the other point to make here is that when one subunit exists by itself and is still capable of being synthesized, it gets degraded very rapidly. So in the cell body, once you synthesize NFL, for example, it rapidly oligomerizes with one or another of the subunits or all of them and gets shot out into the axon. And if it doesn't, it gets degraded unless in a pathologic situation. So we were encouraged by all of these little bits of information to go further and to look at synaptic neurofilaments. Were they present in the synapse? And indeed they were. In a gradient, one sees a vesicular fraction in the gradient that is highly enriched in neurofilament proteins. And this, surprisingly, in cortex and hippocampus, represents about 25% of the total neurofilaments in, and it starts to explain why you do see neurofilaments in the form of, or decorating axons, but it's very much more limited than what you see in the white matter tracks. And what we realize is that in cortex and hippocampus and those areas that are very cellular, most of, a large proportion of the neurofilament is in synapses. And it's different in its composition of, or the stoichiometry of the subunits. These changes in the ratios of one subunit to the other, or the phosphorylation of the H and M subunit, are different than what they are in the total pool. And this shows that if you knock out M subunit, that you change the total pool differently than you change the synaptic pool, so that these pools are being modulated to different extents depending upon the conditions. And we have now many more examples of how independently they can be altered. We started to look more precisely at where these subunits were, and in cultured neurons you could demonstrate co-localization in postsynaptic sites in relation to PSD95, but also in presynaptic sites as well. And this was kind of the first indication that the neurofilaments are at both ends of the synapse, but not a very good resolution. So we started to look more deeply ultrastructurally, and this was an area that if you looked in the literature with with the prejudice that neurofilaments were not in synapses, people would see filamentous structures and say, well, that has to be actin, or that has to be something else, or there's just there's only microtubules. And it really took an effort. And in fact, this is a human brain where, ironically, we were able to see the best ultrastructure. It happened to be a biopsy, and we were grateful for the graduate student that volunteered for this study. But just kidding. I realize that we're being videotaped here, so anyway, but biopsy human brain, you could see these structures, which at this stage they are 10 nanometer filaments, but they're obviously very different from neurofilament that you see in an axon. Short, tortuous, and not very linear. And in a mouse, you could label with various antibodies. In this case, here's NFL and H together, and you see on a single linear structure in the synapse a decoration by both the antibodies. And we did the very tedious quantitation of the EMs and concluded that that there is a greater abundance of neurofilament in the postsynaptic aspect of the synapse than in the pre-terminal aspect of the dendrite. So dendrites are not known to be super abundant in having neurofilaments, but they do in fact have neurofilaments. But there's an enrichment in the postsynaptic density, and the presynapse also has presynaptic element also has a significant amount over what you see in the neuropil. So there's enrichment on both sides. And this study came out just a month ago, confirming what we showed in a somewhat different way, using antibodies and synaptic markers, and concluding that indeed the postsynaptic region has a higher localization than the presynaptic region, and that there's a quantitative difference in synapses of different types, and that this is probably not going to be surprising in the long run, that synapses, if they're functionally serving different roles, will have different stoichiometries of subunits in different types of synapses. But it was nice to see this confirmation of the co-localization at the immunocytochemical level. So without going into detail on this study, this is really some of the evidence that neurofilaments are in synapses was in this paper in Molecular Psychiatry in 2015, and work mostly by David. And it was known at the time that Michael Ehlers and Richard Hugenair had 20 years before published a paper saying that NFL binds to the NR1 or the glue N1 subunit, period. You know, interesting observation, but not very much context as to what this meant. But it was enough for us now to start to use that information to dig deeper. And as I say, I won't go into it. main headline, though, is that when you knock out M, you create a hyperactivation of the D1 receptor that makes the animals look almost as though they're chronically on cocaine. And so we're trying to form a company to, you know, again, I'm kidding. But the thing that is remarkable and was remarkable at the time is that that you could, in fact, knock out a subunit, and you would have some changes in the other subunits, relatively minor, but you could get phenotypes that would be specific for that, that particular subunit. So again, even though we think of subunits as being assembled into these neurofilaments, and if you knock one out, you throw off the axon in various ways. In the synapse, you can knock them out in the mouse brain, and in the synapse, they will have very selective effects on behavior. And you probably can't read this, but these are various knockouts, and you can see the fact that the M knockout, as far as locomotor behavior, is unique in its effect in hyper-activating locomotor responses. And this is the knockout. Here is the heterozygote. So having 50% M was enough to restore normal locomotor activity. And we went a lot deeper into the mechanism and were able to support the idea that when the D1 receptor is on the surface, the postsynaptic surface, in response to a stimulus, it can be desensitize the synapse by internalizing in endosomes that then bind to these assemblies that contain NFM. If you don't have the NFM in the assembly, it doesn't bind, and it just shuttles immediately back to the postsynaptic membrane, and that's what gives you this D1 hyperactivation that looks like you've overstimulated the receptor. And we were able to demonstrate this model in cultured neurons and brain. So moving to the NFL and its relation to NMDA. Again, here's the receptor, and what was known at the time is that the receptor is regulated in part by ubiquitination, which stimulates endocytosis and degradation in either the proteasome or the lysosome. And armed with that information, I'll just, without going into the details of these outcome measures, we isolated synaptosomes, and just to point out, there's no L. There are changes in some of the other subunits, but with respect to M and internexin, those changes are not enough to move the phenotype. And our evidence is such that having a little bit of H is enough to maintain that phenotype as well. So the main effect is totally removing NFL, and we were able to show that it supports the structure of spines, that they become reduced in length, and actually some disappear. And this correlates and is probably related to the finding that LTP is significantly altered. I should point out that with the M knockout, that LTP is not affected. So not every subunit alters LTP. H knockout and internexin knockout do in fact alter LTP as well. We did binding studies and show in both directions that NFL binds the GLU-N1 subunit, and that in fact using a ligand to the surface GLU-N1, it's significantly reduced, more than 50%, and at the ultrastructural level we could see that this was especially apparent in the synaptic region as opposed to extrasynaptic locations, and that we could measure the amount of lowering by Western blot. And related back to this observation that ubiquitination is what increases endocytosis of the receptor, the loss of NFL increased the ubiquitination and presumably increases the turnover in endocytosis, which would account for the reduced levels of the receptor. This, I threw it in because it made the psychiatrists who reviewed this delirious for some reason, because we used imaging to show that glutamate levels by spectroscopy are increased, which is what you would expect if there's hypofunction of the NMDA reception. This is very curious because the NFL knockout is, as I mentioned this morning, their mutations have a very severe effect, but that the knockout has a relatively mild effect on baseline function in terms of strength and muscle strength, as indicated by grip strength. And when we saw this profound effect on locomotor activity, we thought, well, that's consistent with just the fact that they have very weak peripheral nervous function. But then if you stimulate dopamine-mediated locomotor activity, they actually have normal capability to move. They're just not feeling like they want to move, if I can interpret psychiatrically what their level of mood is. On the other hand, PCP, which is an NMDA-mediated locomotor activity, is profoundly reduced. So motor activity specifically related to NMDA function was profoundly reduced. And as one might expect, all of the NMDA behavioral functions that are known, have been reported, are all impaired in these mice. And this, I think, is probably the most interesting part of the story, is that even a 50% reduction in NFL is enough to create this hypo-NMDA phenotype. And the reason that this is interesting is because in certain regions of the brain in schizophrenia, there are reductions of NFL that are in the range of 50% reduction. And in Alzheimer's disease, there are also reductions in NFL, but also in a number of neuropsychiatric diseases, there are alterations in And this, again, are observations that were made by multiple people over time and have been, the schizophrenia work has been confirmed many times. This is a really robust alteration in schizophrenia. And that these are levels of reduction in these mice that are comparable to what is seen in some of these diseases. So we really think that this is a meaningful insight into what neurofilament changes at synapses could imply for neuropsychiatric disease. And finally, in just a couple of minutes, Jean-Pierre already described neurofilament turnover in general, and this is a summary of the mechanisms as we've interpreted them over the decades that we've worked on transport of neurofilaments. And there is a component of transport of neurofilaments and also of shorter oligomers and as small as heterodimers that is moving at a rate that's faster than the neurofilaments that might be assembled are moving, and those are coming on and off of a fast transport mechanism. And some people call them paused and then moving. And a similar type of concept relates to assembled filaments, which are probably short, not the ones that you see in axons that are very large and are actually stationary. That these are moving, they may be paused for a while, but then they are precursors to a stationary lattice that is extensively cross-linked. And that, from work that we've done in several studies, and Jean-Pierre has also shown that lattice turns over very slowly in axons. We measured about 53 days back in 1986, and Jean-Pierre was 3 to 4 weeks. So remarkably, we came to a similar conclusion, and that the gist of this is that what is actually transported at any given time may be precursors and may be a very small pool compared to what's actually in the axon. And that's why energetically neurons don't die out after one day trying to synthesize all this neurofilament. They're only synthesizing a small pool which moves down, gets incorporated into the stationary cytoskeleton, or it might reach the synapse and then function there before being degraded. And this, I think, second to last slide is a very broad summary, just to make a few points. This is work by Malarao in triple transfected SW13 cells that have no neurofilaments, no neurofilaments. You can transfect and form neurofilaments very easily in these cells, and then you can look at the turnover. And something that had already been known, calpain is a very active potentially to neurofilaments. But what we were able to conclude from these studies and from in vivo studies is that lysosomal degradation was an active process in these cells, and also for at least 3 of the subunits in vivo, when we used an inhibitor, 3MA, to inhibit autophagy, and in this case, 3MA, pepstatin, a protease inhibitor, kanamycin A, a proton pump inhibitor, all of these lysosomal-related mechanisms of of degradation. Surprisingly, MG115, not the end-all for proteasome inhibitors, was not as effective in altering levels. And it's not to say that it's not a player, but I think the conclusion that we're drawing is that autophagy is a significant player in the turnover of neurofilaments. And there are some implications that might relate to the release of fragments and things that would be of interest to, without actually killing the neurons or degenerating the neurons, you can get exosomal and exacidic release of various autophagic and lysosomal fragments. So if they're being degraded there, and this is to drive home the point that in fact this is in vivo, very obvious co-localization of NFM with LC3, an autophagosome marker, and cathepsin D, a lysosomal marker. A surprising degree of baseline turnover of neurofilament in cell bodies, which may explain why there isn't a lot of, normally a lot of baseline neurofilament in the pericardia of neurons. And conversely, if you are impairing autophagy or upregulating autophagy, you could have significant implications for releasing, generating various fragments that would be relevant to biomarker. So these are the conclusions. Neurofilaments surprisingly abundant in synapses. They're distinctive, especially in terms of individual functions of the subunits as well as their structure. NFL in particular binds to the NMDA receptor subunit, and it regulates its activity. I didn't show this, but I explained that neurofilaments are turned over locally along the axon as opposed to one particular site, and they have different susceptibility to proteolytic systems, which could be interesting in terms of how certain metabolites might appear for a given subunit and not for another. That once they disassemble or are picked off of an assembled neurofilament, they have different routes of metabolism. So again, I mentioned the key collaborators, long-term collaborators for this work, and Jean-Pierre, of course, for his long-standing collaborations. So thanks.
Thank you very much. Questions? Yes.
You want me to take it to him?
Thank you for a very fascinating talk. I'm really intrigued by the data you showed, and the in vivo data, where you showed the 50% reduction in NFL levels in relation to synaptic plasticity. I wondered what stage in the disease process the animals you used were, and how you would relate it to progressive neurodegenerative diseases like Alzheimer's disease. So, for example, would you expect during the prodromal preclinical stages where you can still try to salvage synaptic plasticity in humans, would you expect to see are there any ways of looking at NFL levels and seeing how perhaps a reduction in NFL levels through whatever compounds are being developed to kind of rescue LTP, for instance? And would you expect to see a reduction in NFL levels and how would that correlate with looking at synaptic plasticity in humans? So what stage, so the question's twofold, what stage were the animals that you used?
Okay, these are wild-type animals that have that are heterozygous for the gene for NFL. So they're essentially, they're not disease models, they're just a mechanism genetically to reduce the levels by 50%. And so we produce this hypo NMDA phenotype, which mimics hypo NMDA phenotypes in Alzheimer's and schizophrenia. You know, I would just to simply and shortly answer the rest of the question. There may be separate regulation of neurofilament subunits at synapses, which may be possible to modulate in a way that wouldn't necessarily require that you know, find a drug that will increase NFL.
Okay. So there's no pharmacological modulator in development that could reduce NFL levels, perhaps at the pre-symptomatic stage. Is that what you were saying? Yeah,
that there are just take the specific example this morning of TDP-43 that regulates the splicing of. We think that it probably regulates the splicing of NFL as well as the other subunits at synapses. So you modulate, as Jean-Pierre did with a drug. Does it also have the same effect at the synapse or not?
Yes.
Can you more specifically affect the synapse, et cetera, et cetera?
Okay, thank you.
So with the M and L subunit, you nicely show the effect on the postsynaptic compartment and the hyperactivity for the dopamine receptor. Do you think you could have a dual effect cooperation from the presynaptic segment?
Oh, sure. Yeah, I mean, to implicate one is not to exclude the other. It's just that the receptors are on the postsynaptic side. They can be regulated presynaptically, of course, but so there would be options to regulate them differently, and whether neurofilaments presynaptically regulate those mechanisms that impact on postsynaptic receptors, that's something we haven't done.
Along that line, when you showed your EM, it looked like there were vesicles associated with the intermediate filaments. Whether that was optical illusion or just
no, that was intent. That was well, they were colored with the intention of pointing out endosome structures, which is what the expected cargo carrier for the receptors being internalized into the postsynaptic.
Okay.
Yeah, that was postsynaptic. And
along the line of the terminal, Benoit Gentil, who worked with both Jean-Pierre and myself, still does, he found by East2 hybrid that peripherin interacts with CYP30, which is an interacting protein of SNAP25. And whether maybe NFL would do that too, if peripherin does. So there might be something about organizing synaptic vesicles, etc., just as a piece of information.
Oh, yeah. We actually know that that's true from proteomic work that I didn't present here, but that reinforces the fact that the proteome, the interactomes for the different subunits are very different, including some, for your interest, some of the vesicle cargo mechanisms would support what you're saying.
Can I just have you pull those thoughts together a little bit more for those of us who are well outside that space, and talk about what you know about for the structure of intermediate filaments within the synapse. Particularly kind of addressing the issue that, on the one hand, we've heard about the intermediate filaments in the axon as being so stable, so long-lived, but now when we're talking about them in the synapses, which are rapidly evolving over time, there must be very different mechanisms of structure and assembly.
Well, I think the literature has well supported the idea that the stability of the axonal neurofilament lattice is due to the cross arms of the H and M highly phosphorylated cross arms and cross-linking proteins, and that if you disrupt that structure, and we've done that, Malarao has removed the tails, you lose 90% of the axonal lattice simply by, and we know that that's because it gets turned over very rapidly. In the synapse, you don't have that cross-linkage. You have no ability to stabilize these assemblies, so they're basically on their own and subject to whatever proteolytic processes, proteasome, autophagosome, etc., that might be present, calpains.
And so do you view these as really modulatory proteins that are binding relatively specifically to the receptors that they modulate without some sort of large quaternary structure that links them together? Or
Well, I think there are two things. One is that a single subunit is capable of interacting with a whole group of cluster of proteins that are part of the scaffold of the postsynaptic assembly, and that you can visualize it either on the basis of a single subunit that's doing this in the case of NFM, which seems to have this very tight association with the D1 receptor, or that a cluster of several proteins, or even a single protein like NFL, interacts with another network that is part of the anchoring scaffold for the NMDA receptor, and that it's not individual subunits floating around. They're participating in a scaffolding mechanism. When you remove one part of the scaffold, in many cases the scaffold breaks down or turns over selectively a receptor that's more specifically bound to it. So.
As I mentioned this morning, I think it was published by Bob Lazzarini and Myers, that a human NFL is able to self-assemble in culture cell, unlike the mouse NFL that requires an FM or an FH. This may change the way in human that neurofilaments move into an axon. NFL is able by itself to self-assemble.
Yeah.
I think it would be possible now with IPS-derived neuron in culture to address these type of questions.
Well, I would just question the plasticity of a cultured cell to be mimicking. We know, just comparing our results to a lot of the preceding results that looked at assembly, the ability of internexin to assemble by itself. It may do that in vitro, but it can't transport unless it's, that is unequivocal, that it can't function along the axon if it can't get in. So assembly is just one part of the story, and I think you have to interpret those data, and especially a human versus something else. I mean, I might be able to get the graduate student to help with that study, but I think he's a little, yeah.
Perhaps I have one question. Do you think synaptic NFL could be the reason for the baseline?
Synaptic what?
NFL could be the reason for the baseline NFL levels we can measure in blood and in CSF. Is that sort of that's a fast-moving pool, and perhaps that's
yes. You know, I can't say anything definitively, but I would say that when you alter one subunit, you don't necessarily depress the other subunits. They can actually be elevated as long as you have a heterodimer to at least stabilize it in the absence of NFL. But we don't have any evidence that NFL levels are increased in NFM knockout. I don't know. You would agree with that?
Yeah, you're right.
Yeah.
okay. Thank you
Thank
very
you.
much. Then it's my pleasure to welcome Roy Weller from Southampton to talk about clearance of proteins and other molecules from the brain. Please, Roy.
And it works. The pointer is this one.
Oh, excellent. Thanks very much.
Well, thank you very much for inviting me to speak today. One of the messages that's come through from the previous speakers, apart from the enormous amount of new information, is the word serendipity, how people have gone on certain pathways. Now, my only connection with neurofilaments was many years ago, and it involved Mike Shalansky when we were working together in Bob Terry's department in the Albert Einstein. And a recent arrival was Henryk Wiśniewski from Poland. And what Henry was doing was injecting aluminium salts over the surface of rabbit brains, and this induced a huge enlargement of the neuronal cell bodies with neurofilaments. Now, Henry was fairly obsessional about his research, and so when he was in Poland, he had prepared these animals, the rabbits, but unfortunately, they were epileptic. And so when he flew from Warsaw to New York with these rabbits, they weren't very well. And when they arrived in London on stop-off on Polish Airlines, the pilot reported to air traffic control that he had 200 passengers and 8 epileptic rabbits on board. So they refused landing to the epileptic rabbits. So they remained on the plane, but unfortunately there was a delay of 3 days at the airport due to fog. So Henrik had to sleep in the lounge at Heathrow and go out every 8 hours to feed and give anticonvulsants to the rabbits. But they got to the United States. So, neurofilaments. This is a familiar diagram, and what I'm going to do is to ask the questions how do neurofilaments get from the brain into the CSF and the blood? We know they're in there because they've been measured. But what is the pathway along those arrows? Well, most tissues in the body have a nice lymphatic supply so that solutes and fluid drain down well-defined lymphatic vessels. But the brain parenchyma has no lymphatic vessels. So how do solutes and fluid get out of the brain? Well, there are 3 major fluids that pass out of the brain. One is blood, and some solutes are absorbed into the blood. There's cerebrospinal fluid that drains to blood through arachnoid villi and granulations, and also drains to lymph nodes along lymphatic vessels. And then there's interstitial fluid that is almost twice as much in volume as CSF in the human brain. And this also drains to lymph nodes, and then, of course, from the lymph nodes into blood. So those are the sort of macroscopic routes. There's also, we heard, uptake of solutes by cells, and also enzymatic degradation and proteasomal degradation of proteins within cells. There's also enzymic degradation of extracellular solutes within the tissue of the CNS. So this is a summary diagram of the various routes of drainage from the CSF. And so we have, from the brain. We have CSF in the subarachnoid spaces and the ventricles, and the CSF drains to lymph nodes through the cribriform plate and through the nasal mucosa down to cervical lymph nodes. It also drains into the blood in humans through arachnoid villi. And there are various other pathways, for example, along peripheral nerves to lymph nodes. The interstitial fluid, however, drains not into the CSF necessarily, but most of it goes along the walls of arteries down to lymph nodes in the neck. And we've called this the intramural perivascular drainage, and for short we've called it IPAD, but with capital letters. So let's examine some of these routes. So most of the research up to now has been the clearance of proteins from the brain in Alzheimer's disease, because in Alzheimer's disease you have a very obvious accumulation of amyloid outside the neurons and tau within the neurons. And also you have amyloid in vessel walls, the cerebral amyloid angiopathy. And if you look at cerebral amyloid angiopathy, what you see is amyloid associated with the basement membranes in capillaries, and also sometimes forming these drusen on the outside. So this is a human capillary. This is a post-mortem specimen. Also you see the amyloid accumulating in the basement membranes between smooth muscle cells. There's a smooth muscle cell nucleus there in this rather banded or ladder-type pattern. And this is a leptomeningeal artery showing the amyloid in the basement membranes of arterial tunica media. And here's a thioflavin showing this ladder-like pattern. So it was suggested from these static images that amyloid was draining from the brain along the walls of capillaries and then along the walls of arteries, deep in the tunica media, in the basement membranes surrounding the smooth muscle cells in the tunica media. And electron microscopy, this is from my late colleague Henryk Wiśniewski many years ago, and what he showed was that if you look at by electron microscopy at the artery wall in cerebral amyloid angiopathy, there's the smooth muscle cell there, and there's the basement membrane between the two smooth muscle cells. And as the amyloid accumulates, the black dots are the immunocytochemistry for amyloid beta 40, and some of the fibrils are accumulating, excuse me, just talk among yourselves. That's what I was hoping. So if the talk gradually improves, you'll know what the... So how these basement membranes form is that there's a contribution from this smooth muscle cell and from this smooth muscle cell, and this line down here is the line of fusion where the extracellular space is actually excluded from the basement membrane. So the amyloid accumulates gradually in between the two layers of basement membrane, gradually becomes larger, and in this confocal image you'll see the amyloid separating the basement membrane. So I'll just make that a bit clearer. There's the amyloid separating the two basement membranes. Now gradually these pathways get blocked by amyloid, and then you get loss of homeostasis in the brain, you get a rise in soluble amyloid in the brain, and this seems to correlate with Alzheimer's disease. So there's loss of homeostasis in Alzheimer's disease. And here's a nice mouse model from Matthias Jucke, and it shows the amyloid all the way along the leptomeningeal arteries and in the penetrating cortical arteries in the walls, and the anterior cerebral and middle cerebral arteries there. So this stimulated a program where we wanted to look at the experimental drainage of fluid and see whether it actually had this pattern of intramural periarterial drainage. So this is the work of Roxana Carrari, and it shows a mouse and the injection of fluorescent tracer into the basal ganglia, into the caudate nucleus here, and then after 5 minutes, which is the time it takes to turn the mouse over to perfuse it, we saw the following, that there was diffusion of tracer within the extracellular spaces, and there was tracer in the vessel walls. And in capillaries, the tracer, which is green, is co-localizing as a blue color with the laminin in the basement membranes of the capillary, just as we saw in the human cerebral amyloid angiopathy. And in the arteries, the blue line is the co-localization of the green tracer with the laminin in the tunica media. I'll just outline that in white. Now the distribution of this is very interesting because you notice that this basement membrane has no tracer in it, and neither does the endothelial basement membrane have any tracer in it. So they are excluded from the outward flow pathways. Furthermore, there is uptake of tracer by cells, possibly an astrocyte or even a neuron there, and certainly by smooth muscle cells in the walls of the arteries. So just as a model, we have amyloid beta or various other traces that we've injected into the brain. They diffuse through the brain parenchyma into the basement membranes of capillaries and then pass along the basement membranes between the smooth muscle cells, but not along the endothelial basement membrane and not along the basement membrane on the outside of the artery. So they go along there and also there, and they also go into perivascular macrophages and into smooth muscle cells. So there's uptake of tracer from and amyloid from these by the cells around the vessels. So to what extent that is a signaling mechanism about brain activity to the smooth muscle cells in the arteries, we haven't managed to test so far. So one of the important things I think about looking at this in relation to the extrusion of neurofilament proteins from the brain along this route is what factors affect the transport of fluid and solutes from the brain. Well, as you get older, there's almost a continuous damage to your arteries, and you get an increase in fibrous scar tissue within the walls of arteries, and also thickening of basement membranes. And so you can show in the mouse that there is a reduction in the efficiency of flow in there's age-related changes in cerebral arteries, and that reduces the efficiency of the outflow. Now, the motive force is obviously very important here, and the latest ideas that we've had, because they're very difficult to test in vivo, is that following the observations radiologically, that there are waves of smooth muscle cell contraction called vasomotion along cerebral arteries, and these waves of vascular smooth muscle cell contraction go in the opposite direction to blood flow, but in the same direction as the flow of fluid and solutes out of the brain. So the best guess at the moment is that vasomotion may have something to do with the extrusion of fluid and solutes out of the brain. And there are clinical trials going on in Japan at the moment looking at stimulating vasomotion with cilostazol, and then there are also plans to look at using apolipoprotein J as a facilitator of drainage of amyloid along these basement membranes, and also looking at stimulating the contraction of the smooth muscle cells by enhancing their nerve supply. There was a very interesting experiment that was performed by Tom Beach in Arizona, and he took some rabbits and he killed off the nucleus basalis of Minot, which supplies the cholinergic supply to blood vessels within the brain. And in these young rabbits, following the destruction of the neurons, they developed cerebral amyloid angiopathy, and then he traced the resprouting of surviving neurons and watched the reinnervation of the blood vessels, and as they became reinnervated, the cerebral amyloid angiopathy resolved. So it suggests that nerve supply may well be an important factor in the transport of amyloid along these pathways. Amyloid angiopathy itself, which can be shown experimentally, inhibits the transport of fluid and solutes along the IPAD pathway. The chemical nature of the protein is quite important. Patients with just needs a regular dose. One of the risk factors for Alzheimer's disease and for cerebral amyloid angiopathy is apolipoprotein E epsilon 4, and Cheryl Hawkes has shown that transgenic mice expressing human apolipoprotein epsilon 4 have a very inefficient drainage pathway, and amyloid beta, of course, is associated with apolipoprotein. So the actual combination of all these proteins is quite important. The upper limit, we haven't actually done these experiments, these are for Nick Barua, suggests that perhaps you can get up to 150 kilodaltons along these pathways. And then Roxana Carrari induced immune complexes in the brain and showed that after 24 hours, the immune complexes were in the basement membranes and they had blocked the perivascular drainage. but after 7 days, the immune complexes had disappeared. And it's probable that if you're using these pathways for the whole of your life, that they repair very ably. Lastly, down here, we looked at perivascular drainage in pups from high fat diet mums in mice. We had noticed that, that the basement membranes in the kidney were greatly thickened in these mice. So we thought that perhaps the same thing was happening in the brain, and indeed it was, and this impeded the drainage of solutes and amyloid. So there are a number of things that impede the drainage en route to the CSF and in the blood. So there are a number of proteins that are known to drain along these pathways. Amyloid beta is the obvious one, transthyretin, and cystatin C in the Icelandic cerebral amyloid angiopathy complex, and ABRI in the Danish and the British type of amyloid. And this also affects the spinal cord. Very little experimental work's been done on the spinal cord with regard to this because of the technical difficulties of injecting tracers into the mouse spinal cord without it leaking straight into the CSF. Now the experimental studies, the very recent experimental studies from Roxana's group, tau passes along the IPAD pathways, so does synuclein, and Martin Jeffrey has been studying PRP. Now PRP is a very, prion proteins are very important because they're released from the cells and they attach to neurons. But if you take off the anchor and inject them, then they just pass out along the IPAD pathways. So this is another aspect. To what extent are neurofilaments attaching to other cells or being taken up by other cells? Now, there are interconnections between the CSF and the brain. So if you inject a tracer, fluorescent tracer, into the CSF, the tracer comes down along the basement membranes right on the outside of the artery. And there is no perivascular space around arteries as they enter the cortex. Milton Brightman pointed out in 1964 that perivascular spaces around cortical arteries are all artifact. But the myth persists, but there is no perivascular space. So it comes down, they come down a basement membrane here, and it's co-localizing with the laminin on the surface here. And by electron microscopy, you can show that here's the artery wall, there's the lumen there, the endothelial cells, and then the basement membranes between on the outside of the artery. And here's the glia limitans, and the tracer, in this case nanoparticles, is in the basement membrane between the pia mater covering the vessel and the glia limitans, so pial glial basement membrane. And then 30 minutes, this is after 5 minutes, 30 minutes later, you see the tracer coming out of the brain in the IPAD pathways. So it comes out along the walls of the arteries, just as if you'd injected the tracer into the brain. And this has been shown by Roxana's lab, by Robert Thorne's lab, Lydia Sorokin, and this is another one of our papers. So the entry from the CSF is along basement membranes, and the exit is along capillary basement membranes and out along the IPAD pathways, as shown here. used to have some sound with it, which was a great cheer as it came out. I cut it out. So here in the diagram, this basement membrane on the outside of the artery has a characteristic alpha-2 laminin. So you can actually show that the tracer and the alpha-2 specific alpha-2 laminin are co-localizing, that it's actually in the basement membrane because there is no space. And here's the human. This is a human cerebral artery, not from a graduate student. And here you see the lumen of the artery here. The endothelium is green, and then the smooth muscle cells are purple, the basement membranes are yellow. And as you go out here, you'll see the pia mater, which is this starred area, which goes down into the brain alongside the artery, and then a basement membrane between the glia limitans and here. So the blue line is where CSF enters the brain, and the red line is the line where solutes and fluid come out of the brain. And you notice that although this is very superficial, this is really quite deep within the tunica media. And as the arteries get larger, then this pathway remains buried in the tunica media. So when the pathway goes out into the leptomeningeal arteries, it's separated from the CSF by quite a substantial muscle layer. So that just gives it in. Yeah. So the big question here is, how do neurofilament proteins that are in the extracellular space, in interstitial fluid, entering the basement membranes, we only have this supposition because we haven't done the experiments to show, but if it follows the same pattern as, say, tau or amyloid beta, then it will go along the basement membranes of capillaries, bury itself in the tunica media, and then go down to the lymph nodes. So how does it get into the CSF? And we know that it's in the CSF, and what proportion of protein drains into the CSF? Well, there have been some studies trying to measure how much solute injected into the brain gets into the CSF. And the very early studies, and probably the best studies, were by Helen Sir and her group in the 1980s. And yes, this is Setsivani and Sir. And what they did was to inject very absolutely minute amounts of radioactive human serum albumin into the basal ganglia of rats. And then they traced the radioactivity, and it went out along the arteries all the way down the neck to the cervical lymph nodes. And where the artery went below the cervical lymph nodes, there was no label left in the artery, which suggests the label was going almost totally out into the lymph nodes. And they estimated from measuring CSF in the cisterna magna that about 10 to 15% of albumin leaked into the CSF. So really quite a small amount. And if the if neurofilament light does the same, it may only be 10 or 15% leaks in. And more modern studies by Guizot, he was interested in whether the oligomers of A-beta moved into the CSF with the same efficiency as monomeric. And he found that monomeric A-beta had this sort of transfer into the CSF, whereas for oligomeric it was only about 4%. So it depends on molecular size. And Dean and Shibata found similar results. Now, one of the problems is the technique of injecting tracers into the brain. And Roxana found this while she was doing her studies and developing her techniques. She injects 0.5 microliters through a cannula that's 50 micro in diameter into the basal ganglia of a mouse in over 2 minutes. And she found if she did that, none of the tracer leaked into the CSF. And if you look through the literature in the last 20 years of people injecting tracers into the brain, they go as high as 2 microliters in the mouse. They inject it very quickly, and then they say, oh, the drainage of interstitial fluid is straight into the CSF, and then into lymphatics in the dura. So a lot of artifact has crept in here. And so the conclusions here is that about 10 to 50% low molecular weight soluble proteins pass from the from the brain into the CSF. So as a conclusion, or confusion, here I think might be the total picture. Here are the neurofilaments released into the extracellular space, and the standard pathway is to go through, diffuse through the extracellular space into the basement membranes of capillaries, into the basement membranes between the smooth muscle cells, and then down to the cervical lymph nodes. And of course, the changes that take place in this pathway, if we look at what happens as it goes through the extracellular space, in younger animals it may be taken up by neurons, may be taken up by microglia, astrocytes, may be degraded by enzymes in the extracellular space. It may also be absorbed into the blood as amyloid is. Then as you get older, there are the changes in the arterial walls that inhibit the transport along this pathway. So you get deposition of amyloid in the walls, and you get loss of homeostasis in Alzheimer's disease with inadequate drainage, changes in the chemical composition of the extracellular fluid, with a rise in soluble amyloid and a rise in oligomeric amyloid. And you also get deposition of amyloid plaques in the brain parenchyma. And the amyloid plaques are like sponges. If you inject anything into the brain in a mouse that has amyloid plaques, a large amount goes into the amyloid plaques. It will absorb anything. And the same in amyloid angiopathy. If you inject a tracer into the brain, and it is absorbed by the amyloid. This has led in the past to all sorts of stories about zinc being deposited in the brain in Alzheimer's disease. They absorb everything. So this again is another problem. Now, looking at human brains with Alzheimer's disease, there is some indication that once you start blocking all these pathways, that the solutes actually drain straight out through the surface of the brain, because you see amyloid deposited in the glia limitans, which suggests it may have gone out there. So the pathways change with age, they change drastically with Alzheimer's disease. And so I leave you with that thought, and just a historical document that in the past it was thought that there was a free flow of fluid and solutes into the brain, out along perivascular spaces that don't exist, into the CSF that acted like a sink, so that the brain didn't have any organized structure. But we know now that the movement of fluid into the brain is very highly controlled by the blood-brain barrier, and also from the CSF along basement membranes. And we know that there's control in passing through the brain, particularly through grey matter by diffusion, through the restricted extracellular space in the grey matter. there's much freer movement through the white matter. And then leaving the brain, the main exit route is along 100 to 150 nanometer wide basement membranes deep within the tunica media and not in direct communication with the CSF. So the future problems, the mouse brain and the rat brain differ considerably in their size and shape and volume from human. So the mouse studies have limited relevance because we don't actually know what the dynamics are in the human, although they do give the general principles. I point out this problem with many of the experimental studies of leakage of tracer into the CSF, so we need to have more active control of injection studies. Lastly, it's interesting that the lumbar CSF is the region of acquisition of the CSF, and this is often 1 meter away from the brain. So somebody did suggest that neurofilaments getting into the cerebrospinal fluid from the multiple sclerosis spinal cord could influence the levels. And so we don't know actually what, which levels we're looking at in the lumbar CSF. And so perhaps some of these problems could be solved by using rather larger animals and also using radioactive techniques and histological techniques to trace them. Thank you very much.
Thank you very much, Roy. Are there questions? Yes.
So Roy, while you're in an iconoclastic frame of mind, tell me a little bit about whether you think the subpeel space still exists or ever existed, and also could you tell us a little bit about whether there's any drainage or why not around perivenous routes?
The arteries in different regions of the brain, both in human and in mouse, differ in structure. So as the artery comes down into the brain from the leptomeningeal artery, and most of the arteries come in from the surface, they take with them a sheath of pia mater. The pia mater cuts off the subarachnoid space from the brain. It's reflected up onto the leptomeningeal arteries, so there's no direct connection, although fluid and solutes get through. Red cells don't, macrophages do, they crawl through it. But the pia mater follows the artery down, and there is just a compact layer of basement membranes and cell processes around cortical arteries as they go in. And then as you get into the white matter, instead of 1 layer of pia mater around the artery, you get 2 layers. And as has been shown in patients with cerebral amyloid angiopathy, you can dilate the perivascular space around the arteries in the white matter. And this is used as one of the assessment techniques for cerebral amyloid angiopathy. And then in the basal ganglia, it's well known that you can get very large perivascular spaces. And again, there are 2 layers of pia mater. So it seems to, and in the midbrain. So it seems that the structure of the arteries are different, and you don't get perivascular spaces in the grey matter, but you can get dilated perivascular space in the white matter and in the basal ganglia. Now, if you inject tracer into the brain in APP mice with amyloid and amyloid angiopathy, or into animals with immune complexes in their artery walls, the tracer accumulates around artery walls, but also around the walls of veins. It looks as if it's an overflow mechanism. And if you inject tracers into the CSF, they go into the brain, they come out along the artery walls, but they're also found along the walls of veins. but the continuity of the veins, the mechanisms, the motive force has just not been shown. There is really, I'm afraid to say, no good evidence that there's any drainage along veins.
And the continuum between CSF and that subpial space was always described as being distinct. But do you think that that's real, or is there any evidence really to show that we can consider those as separate fluid compartments?
Sorry, I'm not quite
so
clear.
Between the subpial, but so when a vein pops out and picks up the pia mater, you could imagine there is a space between the pia and the glial limitans, which is often described as a subpial space.
Well, the structure of the veins is different from the arteries, so that the venous perivascular space is expandable, and you see lymphocytes accumulating in perivenous spaces, but not in periarterial, around arteries. And there's no complete layer of pia mater. And so there's a free communication between the subpile space and there's often some small space adjacent to veins, but not arteries. So it's a different structure altogether.
Okay, so that exists, but in terms of connectivity between the subarachnoid space and the pia, do you think that's so that Roy, maybe you don't believe, and maybe this isn't sort of been something you concerned yourself with, but there used to be a school of thought that actually there was sort of another compartment sat in there that was contiguous with that venous space that then didn't drain into the sort of the standard subarachnoid space.
Well, in humans, where everything is so much larger, it's jolly difficult to work it out in the mouse. And what you have is the pia mater is a single layer of very delicate cells, and it comes along and it reflects up onto the walls of the veins, cutting off the subpile space from the subarachnoid space. But it does not go down into the brain alongside the veins. So the subpile space is continuous with any space around the veins. But this is not the case in the arteries, where the pia mater completely cuts it off.
I noticed that all of the proteins, I think all of them that were demonstrated to go through the iPad were proteins that are capable of being oligomeric or to aggregate, and presumably are more difficult to metabolize. Is the fact that that group is solely composed of that type of protein coincidental, that these were proteins of interest in diseases and were looked at, or is there some feature of proteins that might determine whether they would go through that pathway?
Yes, I mean, they were proteins of interest from the point of view of Alzheimer's disease, which is our major focus, and so that the tau and the synuclein to do with Parkinson's disease. And also, of course, they are soluble proteins from the manufacturers. They're not, I don't pretend that they're actually native proteins. Similarly, the amyloid beta is a soluble amyloid beta, and it's not attached to lipoprotein. So we're demonstrating principles. The details are still to be worked out.
So just to follow up that for a second, I mean, these are, many of them are autophagic substrates, and so in terms of how they are getting out into the interstitial fluid to begin with, I wonder if there's some relevance of how do you think they're getting there to be cleared through that mechanism?
Well, it's been more or less demonstrated that with tau, that the progression of tau involvement of neurons is probably by direct spread along pathways, and it's been shown experimentally that it goes into the extracellular space, is probably taken up by adjacent neurons, and then starts the whole process of hyperphosphorylation, etc. Similarly, some transplantation studies, probably by accident, of synuclein-containing cells showed that there was transfer of synuclein from cells to other cells in vivo. And what we showed in the experimental studies was that tau drained more quickly than synuclein. And synuclein was taken up by neurons and by microglia within the extracellular. So there's quite a diversity of how the proteins, even in our sort of fairly simple injection studies, behave within the extracellular space.
So you told us about the factors that will diminish IPAD, and I'm getting a little concerned that my iPad is diminishing as I'm sitting here. Are there any factors known to improve IPAD? Does sleep matter? Does obviously diet? Any hope?
Well, there are good indications that celestazol improves the drainage along IPAD. It certainly does in experimental animals, and there are clinical trials going on at the moment to see whether it reduces cerebral amyloid angiopathy. But I think the problem with these clinical trials may well be that they are treating the patients too late. Somebody mentioned this morning about the trials in Alzheimer's disease and the antibody trials, the anti-A-beta trials, and right from the start, it was recognized that although the A-beta plaques disappeared in these trials, the amount of amyloid angiopathy went up, and also did some of the complications of blocking the IPAD pathway, and that is white matter edema. And in fact, it was stopped because of the white matter edema was mistaken for encephalitis. But a lot of them have white matter hyperintensities following. And in one trial, they lowered the dose and got rid of the white matter hyperintensities, so that if you reduce the rate of destruction of the plaques, then you can reduce the amount of amyloid going into the drainage pathways and regulate it that way. So there seemed to be quite a dynamic pathway. It's not just sort of stopping there. I mean, some of the post-mortem cases of the patients who had PET confirmation of amyloid in their brains, treated with anti-amyloid antibodies, and then shown to have amyloid angiopathy when they died. And then there are other cases that survived longer, where the plaques and the amyloid angiopathy was no longer present. So, I mean, it's only observational post-mortem material, but it could be that this dynamic system could repair itself, could get rid of the amyloid. So I think the future is bright.
So many of intracellular protein aggregates can be secreted via exosomes. The exosome, can they be secreted the same way, the clearance from CSF to the blood the same way?
Sorry, the intact exosomes?
Exosome, yeah, but they sometimes contains aggregated protein.
It would really depend on size. Of course, our entry into lymphatic drainage was really through immunology, because the question is, how does the brain connect to the immune system? And the brain, of course, has immunological privilege, so that no antigen-presenting cells can drain along this pathway directly from the brain. And so whether the vesicles could go along that pathway, we'd have to look at the size, compressibility, etc.
May I just go back to this difficult and important anatomy in the periarterial space? So the drainage in the basement membrane lying between the smooth muscle layers, is that what I just want to understand is why the molecules in iPad are directed in that space particularly? Is it the convection that's set up with the contraction of the smooth muscle cells on either side, or is it because of barrier limitations preventing movement into the related glia limitans?
The short answer is we don't know. But what we have, we collaborate with mathematicians to build models. And the best guess is that the major pathway is in a special part of the basement membrane that effectively is where the extracellular space has disappeared from the apposition of the smooth muscle basement membranes, and that the contraction, serial contraction and directional contraction of the smooth muscle cells is driving it along this particular pathway. And with age you get changes in the basement membranes that might disrupt it, or deposition of amyloid and other insoluble proteins. So that we don't know the details of the physiological mechanism, because they're very difficult to study within the parenchyma of the brain.
The related experiment that you mentioned was this was the degeneration of the nucleus basalis,
Yes.
which is cholinergic.
Yeah.
And obviously a lot of this system is cholinergically
Yes.
driven. So addressing Heather's question in a different way, I mean, is this a highly pharmacologically responsive drainage system, do you believe?
I'm not a physician, but I understand that Aricept is a cholinesterase inhibitor, and it's Tom Beach did some. Actually, he used physostigmine to show that he could actually reduce the amount of amyloid angiopathy in these animals by replacing or enhancing the cholinergic action on the vessels. So it could be that if Aricept has any effect at all, that it could be an encouraging iPad.
Thank you very much. I was thinking if we should take a leg stretcher, but perhaps it's okay now. I will close because of the recordings also. But now you got some fresh air, and then we have coffee. No, actually, we start now. Yeah. So no leg stretcher. We start now. No leg stretcher. Yes. But then we start. It's my pleasure to introduce you, Pascale. And now we will hear about protein degradation. Yes. And Pascale comes from University of Montpellier.
Pascale, you said you wanted the
Yes,
Do you want
thanks.
them all
Yes.
for the duration or just for the movie?
Or for the duration, yeah. So I'm grateful for the invitation. So it's really terrific. You could gather all the different expertise, and I think this is what makes this discussion so interesting. And I learned a lot throughout the day. So I thank you both very much. And I will start apologizing. I have no fancy, spicy story to tell you about the reason why I jumped into the neurofilaments. But instead, maybe my personal touch could be some nice movies that I do live in zebrafish. So I ask to reduce the light so you could relax. Not too much. And my main interest, of course, my favorite protein is neurofilaments. Oh yes. My main interest is neurofilaments and the degradation of this intermediate filament. But before, I will just give you briefly, within a few minutes, an overview of the family of intermediate filaments. Intermediate filaments is a large family comprising many genes that come with different flavors across the tissue in the human bodies. So you have keratin in the hair, lamine is within the nucleus, and desmin in muscles, and for us, most importantly, neurofilaments along with internexin peripherin in the nervous system. So they exhibit a complete distribution and high regulation, not only throughout development, but also in adults during injury and trauma. So mostly all the genes of this family have been implicated in disease, in human pathologies. So the biggest one, I believe, is laminopathy, what we called after mutation found in the lamine genes. You can have lipodystrophy, muscular dystrophy, premature aging with progeria, cardiomyopathy with desmin, and of course, neurofilaments are mutated in a couple of diseases, and specifically Charcot-Marie-Tooth disease, but we heard this morning this predisposition with ALS and Alzheimer. So, of course, the study of all these pathologies in animal models allowed to highlight the functions, the complete function of this cytoskeletal component way beyond structural and mechanical integrity. So we know now that intermediate filaments play crucial roles in gene transcription, axonal transport, but also cell migration and important cellular processes such as cell invasion. So here it's really a mutation in genes, but there are also many other diseases which are characterized by aggregation of intermediate filaments. Myopathies have found a lot of inclusion of desmin, also the same with the Mallory-Deng bodies. Here we are, in the audience, mostly interested in aggregation of neurofilaments, which are seen in most, if not all, neurodegenerative diseases. So while here a specific intermediate filament, when aggregated, can be associated with a specific type of disease, I will talk about a very exotic but interesting disease, which in fact implies all intermediate filaments throughout the bodies. So this is the sum up of what we have done and what we have seen in human, so with the clinicians, and now we'll show you the study on the different animal models. So here the disease is named GAN for giant axonal neuropathy. Here are some of the examples found really early on the patient biopsies. Here, vimentin is very important. I will describe here this model. It's coming from the skin biopsies derived from patients, where you see these ovoid bundles of vimentin. Neurofilaments from the central but also peripheral nervous system. These are the aggregates of desmin, and keratin is, with the severe form, an hallmark of the disease. So interestingly, this disease is really affect massively the central nervous system. It starts early on during infancy with the peripheral nervous system, and as you can see, it's really massive because it leads to a total loss of sensitivity in the teens, and from really 2 years of age child, it spreads through the motor system and leads to a total loss of ambulation. Afterwards, this disease spread to the central nervous system, and you can see here the different impairments. So you have many different kinds. You can have ataxia, nystagmus, optic atrophy, dysarthria, and mental retardation. This is accompanied with the presence of enlarged axons, which are also an hallmark in other neuropathies, such as Charcot-Marie-Tooth disease. And this disease is fatal, and there is actually no treatment for this rare disease. So to me, really early on, these important features that I just described really highlighted this essential role of this gene, the GAN gene, in sustaining cell survival, but also cytoskeleton architecture. So I'm coming from a genetic background, so I was really working together with clinicians on patient samples to do homozygosity mapping, because it's a recessive disease. So we clone the genes, and way beyond the sequencing of the human genome, we play around with bioinformatics to identify the gene, which encodes for a protein we name gigaxonin. to refer to the giant axons, in fact. And here are the first mutation that we found in patients. So the systematic sequencing of the gene allow us to I will come back. Allow us to define the first diagnostic tool for the disease. And here you can see that you have to do that because mutations are scattered along the entire protein. And in fact, in the diagnosis perspective, it's really important because there are too many mutation types, missense, nonsense, deletion, and even the large deletion cannot be directly seen by regular sequencing. So there was really a need to develop a new diagnosis test. And in fact, what we did is that we designed an antibody which was very powerful and showed, I remind you, it's a recessive disease. So these are the lymphoblast cell line derived from patients, where you can see here that we have a highly decrease in abundance of the gigaxonin mutants within the cells. We quantified this, and importantly, we showed that it's really penetrant and specific for GAN. And this is important because GAN resemble forms of Charcot-Marie-Tooth diseases clinically, but also at the histopathological level. And here, so far, we have a very 100% of penetrance of this test. So we are being approached worldwide to use this test. We also wanted to address why we had decreased abundance, and in fact, we did the modeling of the protein to show that in fact all the mutation affect either the folding, the interaction with the dimer, because gigaxonin in fact acts as a dimer, and we found mRNA stability together with protein stability as the cause of this instability. And then I come back to this. So gigaxonin, in fact, is a substrate adapter of a E3 ligase complex, which briefly is targeting the substrate to be ubiquitinated by the enzyme E2. And this is basically the modelization, which is based on the crystal structure of the BTB domain, which is the one that interacts with the complex. And here is the beta proper-like structure from the C-terminal domain, which we call the Kelch domain. So today I will talk about the two research projects we are working on. First, the control of intermediate filament homeostasis by our E3 ligase. And second, we'll talk about the study of neurofilament dynamics in general in health, but also in a couple of neurodegenerative diseases. And for most of them, I will show you a lot of unpublished data. Here is just one slide to sum up a lot of work from our lab, but also Bob Goldman's lab. So we are playing with primary fibroblasts of patients to study vimentin aggregation. You can see here the massive bundling of the vimentin in patients. So we describe this to show that it's highly insoluble, it's conditional, it's really nice because we can increase, decrease, and it's a highly dynamic process. It's really specific to intermediate filaments because the other network look normal, and we even say that microtubules are protective from disaggregation, meaning if we impair microtubule stability, we'll even exacerbate this phenotype. So we were part of a study led by Bob Goldman in Chicago, who showed that, in fact, if you do the opposite, so here it was based on the absence of gigaxonin patient, if you do the opposite and you put a big load of gigaxonin on wild type or patient cells, you have a clearance of vimentin, which is really total. And it's mediated through the interaction of vimentin with the rod domain, which is common to to all intermediate filament type. And while they were not able with this system to demonstrate directly an ubiquitin-dependent degradation of vimentin, the proteasome was implicated, which was really interesting. So to really assess for the role of gigaxonin in neurofilament maintenance, we developed a mouse model and also collaborated with Jean-Pierre, Julien. We were mostly part of one couple of experiments within his study with Florence Desquin. So in our case, we deleted the early exons of the gene, and Jean-Pierre targeted directly the promoter and the first exons. So this was really important to optimize our reagent, and we have now a beautiful antibody where you can see here the specificity of the staining. This is a lot of different tissues, and we have here the knockouts showing the robustness of the reagent. We found that gigaxonin is equally and proportionally expressed within the nervous system, but importantly, and it's nice in regards to the other intermediate filament type, it's also expressed in the muscle, the kidney, the liver. It's also, gigaxonin is also more abundant in prenatal, which is interesting. Of course, we investigated the abundance and the organization of neurofilament within nerves of this mouse model. And here you have the fold increase in comparison to wild-type mouse. And in fact, here we used 3 different mouse models, ours, Jean-Pierre Julien, and another, which was basically mimicking our genetic ablation. And you can see here over time in different tissue, brain, lumbar section of spinal cord, and sciatic nerve, that NFL was the main target with up to 7-fold increase in the knockout mice. Interestingly, we also investigated the spatial orientation along the axons. So we did that in ventral root, dorsal root, and sciatic nerves. It's cross-section, so the way to investigate orientation was to define a circular value, which is in fact correspond to the longitudinal orientation along the nerves. And here you can see here in different spots, the sciatic nerve, ventral and dorsal roots, that you have a decrease in the knockouts, which really reflect the non-circularity, so the longitudinal orientation across the section. So in conclusion, we are working on a rare exotic disease, but which shows interest for the intermediate filament field. It's a E3 ligase adapter per se, which controls the degradation of the entire family. And here I think we have developed many tools from patient samples, the new mouse model now, and the zebrafish that will help to study the live degradation of neurofilament within animals, and also to look at the neurofilament aggregation process in disease. And with that, I finish thanking the member of our team. It's quite a small team. All our collaborators, from clinician to fundamental groups, and especially Jean-Pierre for his support and great interest in the topic. And here it's our funding agency. And I thank you very much.
Thank you very much for a great talk. Questions for Pascale?
Very nice. When you talk about degradation by gigaxonin, are you referring to a mediating protease, or are you proposing that gigaxonin actually has proteolytic activity itself?
As part of E3 ligase complex, gigaxonin drives direct ubiquitination of NFL for sure. We are investigating the other subunit, which leads to degradation. We have to solve out if it's pure proteasome or if it's also autophagy. Or it's also autophagy, because as you mentioned, autophagy is also playing a great role in the clearance.
Right, and I wondered in terms of the also the, is it the I can't remember, the K48 versus the
K63.
63, and one is more likely to go through one of those routes than the other. In this case, what's a ligase adding, 63 or 48?
So in fact, this 48 is more prone to proteasome, and 63 more endocytosis, autophagy pathway. So for that, you have many tools, and we are setting up in the lab the histidine pull down, where you could have like ubiquitin that has been mutated with all the different positions. So then you could solve what is the exact chain, but we are not at this point yet.
Or it might depend on the form of the neurofilament, because if you're doing segments, it's going to be autophagy, makes sense. When you're doing pull downs, you're not really addressing that part so much. You're having more probably soluble oligomers, so that may go through a different pathway. So they aren't mutually exclusive, and it might depend on the situation.
Pascale, may I just go back? Perhaps could you, maybe in conjunction with the others, really try to summarize this, the question of how degradation pathways might be selected, because the endosomal route is potentially so important to think about in terms of extracellular movement of the protein. So what are the factors that might go into sorting for intermediate filaments between these two degradation pathways under different conditions? Can you kind of pull this all together for
Can
us?
I talk about more general intermediate filament? So far, in the field of intermediate filaments, proteasome E3 ligase is the most common element. For autophagy, really exact autophagy involvement in the clearance of IF, it's been only shown for lamine in a great paper published in Nature like a few years ago, but it's in certain circumstances. For example, here it was in a system where it was a cancer treatment. It was around resistant to some treatment, some senescence in cancer. And only here at this moment they saw that the nuclear lamina will undergo nucleophagy through blebbing of the nuclear membrane, which was very impressive. But beside this specific event in response to a specific treatment, nothing is known about lamine degradation. And for the other intermediate filaments, mostly proteasome is a
So that I mean, that's certainly what I thought, from the literature too. But let's just go back and look at the synaptic intermediate filaments. Do we think that those are degraded by the same sort of pathway? Because again, there's another wrinkle in terms of the way synaptic proteins may be processed.
But I have the same same effect that I didn't show, that the subunit respond differently to drugs. I have to check out if it's in the same direction, but the subunits are not degraded through the same pathways.
They are
not degraded.
No, they're not degraded.
Yeah. Similarly.
Right, they're degraded in a different mechanism. Yeah. Which, do we know much about that?
Well, I wouldn't necessarily, at least I didn't intend to imply that they have a different degradative route. In fact, the proteolytic data that I showed is not related to synaptosomes or to synaptic, it's to brain. And if I were a betting person, I would not think that they're degraded that differently in the cell body versus the synapse. I think the issue is the assembly state, number one, and that if you've got an assembled neurofilament, it's not going to be a substrate for proteasome. If you have an aggregate, it's not going to be, and I would even wonder whether NFH, with its tail phosphorylated, can get into a proteasome channel. So I would say as subunits that they're all potentially susceptible to a proteasome or to autophagy. We actually see more activity in the autophagy pathway based on our inhibitor studies. As I said, I don't think we've at all ruled out an equal or greater activity in the proteasome. And the other issue is that I think if you're inhibiting one of the systems, you probably are upregulating the other system, and that they're working in partnership. And the final thing is that once you break down the holoprotein, the metabolites are actually degraded by maybe a different protease than the holoprotein was. So there's a complexity to the metabolic products as to what route of metabolism they...
Yeah, but as we discussed about subunit or filaments, it's also a limitation of the techniques which enables to see which pathway could be affected. Because here, if you do an EM, and I think it's what Raumala did with his tailless mice, he showed us at some point that you have the engulfment of membrane, which is characteristic of the autophagosome. So this, when you see that at the EM level, you know that it's happening. But for proteasome, you don't see, you have no marker at this level that could tell, the proteasome is acting. So it's also limitation of the technical tool.
And the other player are calpains, is calpains. So at areas where you have calcium influx, LDP, presynaptic terminals, injury, various disease things, you could be, calpains are going to cleave, and then what happens to those cleavage products? So it's even worse.
Right.
It's how to know where it's complex.
Right. And the calpains can only degrade in a limited fashion, so that you are obligated to involve some other proteolytic systems once you cleave with the calpain. But they may be the most important in initiating.
And it can also be multiple aspects, because gigaxin, for example, we have no idea at which step of the filament formation it makes the job. It could be after it's been chopped by calpain or...
I don't remember, but the zebrafish, do they have the NFH?
No.
They don't have NFH, NFL, NFM,
Oui.
internexin or equivalent?
Yes, they do.
have this?
Yeah.
Okay, if there are no more questions, we thank you again,